So that's kind of a cool thing. All right. So again, on Monday, when I have my imaging system, I'll be able to show you some of these colored images. But be sure to study those figures in your textbook. A very helpful way to learn this process and form mental pictures of these pathways and how they interconnect.
Are you going to do a review on Monday, or is it going to be lecture on something
Well,
else?
I can take some questions on Monday. I'm not going to just go back and repeat in that terms of a review. But if you'll come with some directed questions, I would be glad to go with that. And I also would love to take some questions now while you've had a chance to see this material today. So give me your best shot. We had a lot of questions on Friday, but we had 33 people there. So if we get a couple students to get the ball rolling here. Janine, you're not shy.
No, but my head's a little full. So basically though, I mean, because I'm understanding this process, I'm not necessarily remembering everything that comes out of every cycle, but we need to know that, right? for the
Well, as
exam?
it relates to the review sheet, what you need to know generally
Well, it
is what
asks
goes in and what
products.
comes out.
Yeah,
That really tells you the whole reason for being for that pathway, doesn't it? If you know what goes in and what comes out, you've got a good handle on what's the purpose. And so when you hear a term like glycolysis, that name gives you a clue. The Krebs cycle doesn't so much give you a clue. But glycogenolysis, lipolysis, even deamination gives you a huge clue. What specimen has amino groups? So there's a lot of clues there for you folks. If you look for them, I think you'll find them. As you're learning them, it will make it a lot easier.
so, but, okay, because one of the questions I believe asked for reactants and products.
Yes.
Okay, so, but I was asking Helen about this. It's like, okay, is the NAD, doesn't to me seem like it'd be a reactant. It's like glucose would be a reactant. 'cause
NAD
it's
is a
going in-
reactant.
It is a reactant.
ADP could be a reactant.
Okay,
So
so
let's do-
anything that's used
Yes,
that's going to interact with another
that's right.
substance.
So let's think about glycolysis. Let's just do glycolysis. I'm not going to do all of them here today.
Okay.
But by doing glycolysis, we can get the sense of it. So we know that on the left-hand side, we have to have glucose, don't we?
Right.
And we already know what comes out. We said that 2-pyruvic acid comes out. And we also said that 2 ATP came out, and that 2 NADH came out, plus 2H plus. Now,
Right.
if I've got ATP on the right side, what must I have on the left side? ADP and PI. So I need 2 ADP plus 2 PI. The PI is the inorganic, non-high energy phosphate group that we're going to attach. If I have 2 NADH, the reduced form, on the right side, I must have what?
The oxidized.
The oxidized form over on the left side, 2 NAD plus. All right? So this really gives us a full accounting, and now we can see, but again, if you know this is over here, surely you know it doesn't appear from nowhere,
Right.
right? It doesn't appear from nowhere. So we have to have the corresponding oxidized form here. We have the high energy form of ATP over here. We have the lower energy form over here. So that's kind of how this shakes out. If we were to do the pre-Krebs, we would do it in the same way, wouldn't we? That is NAD oxidized on the left side, NAD reduced on the right side. If you were to do this in detail, the one we just did, as a chemist did, you would actually put 2 ATP here, plus 4 ADP, plus 4 PI, 2 ADP over here. But I've tried to simplify it, and we're ignoring that. Because for glycolysis to go, you have to put in 2 ATP, and you actually get out 4 ATP. That's the reality of it. But we've
Right.
simplified it, and what I want you to know is, what are we getting out of it? By learning this, I think it's pretty easy to extrapolate back to these, just to balance the equation. Anybody else want to help Janine out? So she doesn't have to carry all the questions.
I don't know how to answer the questions.
Oh, come on, just think, Abby.
I am.
Here's the chance, guys.
And we're welcome to invite questions from our non-enrolled members too. We have 3 non-enrollees. That's pretty impressive. I hardly ever get 3 visitors. I mean, to get 3 visitors on a Saturday? You must have known the cookies were here or something.
So
Or
without,
I was going to sing a song, right? That's
And
why
it's
you
called,
came.
you know, another person to help,
Oh, we've got a question?
so without insulin,
Yes.
something doesn't break down into, I don't have the right terminology. Bodybuilding, they
Yeah.
become skinny.
That's right. What happens without insulin? Without insulin, we can't move the glucose into the cells. So we have it in our diet. It stays in the blood. We have high blood level of glucose, but the cells can't access it. So these people, to get ATP, what are they going to do if they can't put this into the cell? This is a non-starter for them. But they still need to do this to make ATP. So they grab this, don't they? They grab this. And then that's where they're going to get their ATP. But that glucose in their blood ends up being somewhat wasted. It also causes harm. But ignoring that for the moment, the glucose spills over into the urine. And so that was diabetes mellitus, literally means overflow honey. Because people learned, I guess they went around tasting people's urine, they really did. And if it was sweet, that's how they diagnosed it before they had dipsticks. You know? So I don't think they drank it, they probably tasted it and spit it out like some people do wine. But that was it, because you have so much glucose in your blood, so much glucose is filtered into the filtrate in the kidneys. In all of us, we have glucose transporters to bring back that glucose that's filtered. But they get swamped in diabetics. So they're pulling back the glucose into the blood from the filtrate, but so much of it stays in the filtrate, it just ends up in the urine. So that's
And then the thirst comes in.
and then the thirst comes in because that glucose is also holding water in the urine. So they produce more urine, and then they have the thirst. Because, as I said before, comparing glucose to glycogen, glucose affects the tonicity, and it draws water to itself. So it's going to hold water around it as an important solute. So it has enormous complications in that respect. Yeah.
So because everything you read, it says, you know, if the body really likes to work off the carbs for the ATP. So if you're denying the body of the carbs for a long period of time and it's pulling off of the fats
Correct.
and the proteins, are there any effects from that, or?
Well, one of the effects I would say off the top of my head is that some of the foods that we would normally eat that provide carbohydrates, things like fruits, they provide other things for us, don't they? They provide fiber, they provide vitamins and so forth. So you're not, and maybe some minerals, so you're not going to obtain those from those foods. So if you watch that. On the other hand, I think you also have to watch out that if you have a high protein diet, that the protein level is high, and I think I don't have it, but remember the oxidative deamination? We had amino acid, oxidative deamination, we produced a keto acid, which can go in these places, plus an ammonia. Well, what happens to this? I didn't